Thank you, Kerstin, and all, organizing such a nice meeting. And my talk will be infectious disease. It's a kind of different than others, but what we tried here is can we apply and can we understand how virus-host infection model, histone interaction, DNA methylation can be visualized, or can we some way map them, and then when it happens and so on. So I changed a bit of title of my talk because I was expecting I registered very early, and then my title was different. And actually what you will see is I defined epigenetic, but it's not yet, just I'm defining methods first. And this was what you will see is spatial genomic rearrangement in influenza infection. And as you all know, what we're talking about is epigenetic and DNA methylation and histone modification. We will focus on this talk. And why it's important for us as an infection biologist, because it's also in attention that targeting infection epigenome is a kind of therapeutic purposes. So many fields are interested on this, but making the model and the observational methods are quite tricky and time consuming. And also, is there any literatural epigenetic regulation defined in viruses? Yes, there is some, especially HCV and then HPV and then HSV. This kind of special latent viruses, they worked quite okay, but it's not conclusive yet, but there is really ongoing research, this is the list. So also, I put this paper targeting cancer epigenome for therapy because this field is more developed. So the methods they use is we can copy to infection biology to model how they regulate the epigenome status. So in this talk, I will briefly give you one example of the technique that we try to develop in influenza virus infection model in vitro, and quickly go through influenza viruses. Like they have negative sense RNA viruses, and they have 3 different pathogenic genera, A, B, and C. Mainly we deal with A, and they both undergo antigenic drift and shift. So this causing epidemics and sometimes pandemics, and we all know some of them already very important. This is actually this is why how they quickly do antigenic shift and drift. The available strategies are becoming inefficient very quickly. So actually all these genomic details, like they have 11 proteins, and mainly they hemagglutinin, the surface proteins are changing, then they change the target quickly, and then they act as a smart way to infect us. And this is much details. My rationale is here is how these viruses are modulating our epigenetics, and how it can affect the designing and disease prediction strategy, designing therapeutics, and so on. And I will only present today the high-resolution immunofluorescence microscopy today, and also in incorporating this study, we did drug screening. We briefly, I tell you that drug screening, available epigenetic drugs are, I made a library of 80 epigenetic drugs available on the plate-based system, and then I screen infection, and then I screen the titer of viruses, if they go up and down, and then how they get affected. And also, I use protein-protein interaction systems, host histone, and the viral candidates. As I mentioned, there's 11 viral genes, and mainly I tested polymerase complex proteins with histone if they are interacting in vitro systems to see which is the strong interaction, where can we start to look for, and then also ChIP-seq analysis and alternatives. And ChIP-seq I didn't run in these samples, but I did cut and run samples on both influenza and RSV samples. But today the example is microscopy. And microscopy part, the aim was observing histones and DNA modifications. I tested how I started. I find some histone marks, like having antibodies. I tested quite some, more than 40 antibodies that if they give observable signal or efficiently labeling, and then 4 out of this library worked acceptable level, but also 2 antibodies for DNA methylation. I will give the example at the end. And this was H3K9 trimethylation, H3K9 acetylation, H4K20 trimethylation, and H2AK119 ubiquinylation marks. So, for example, what we do, we infect the cells with the high MOI virus. Like at the same time, all the cells are infected. We are assuring that using MOI 10, for example. And by doing this, every hour I take the samples like 0, 1, 2, 3, 4, 5, 9, and then until the one virus circle. So then we label the virus and we label the histones at the same time. And then in this example, you could not see the merge file because the antibodies were not compatible to each other. Later we solved this problem, but in this one not. But we knew that after first hour of infection, virus reached the nucleus, and then up to 3 hours they do interesting modulations, and then later it goes back original state. So actually I pass quickly. This is how it looks when you infect the cells in the control and nucleus, and then mock infection. Mock means just we apply the virus, but we stopped at the same time, just less than a minute exposure, and 1 hour and 3 hours, and the other hours were getting back to normal control level, so we exclude the figures. First example is H1N1 in A549 cells, lung cells, and this trimethylation repressive mark we could label successfully, and they have a special pattern. Here you can see this small dots like pattern is a well-known H2K9 trimethylation repressive mark pattern, and then we use a method to quantify these different samples, and we clearly see after first hour of infection there's clear signal increase, which this intensity show us there is a reasonable regulation in this hour, and then later on it goes back to control levels almost. And another mark we did, this was also a kind of acceptable level of labeling, and then same happened. This is H3K9 acetylation permissive mark. So this was also in the first hour, both acetylation and methylation, most probably in different part of the genome. And another marker, I could see good labeling examples and no effect, because this was H4K20 trimethylation repressive mark. One of the best antibody combination worked for us, but we can see here that the modulation is not that obvious like acetylation and methylation here in the H4K20. And last example I want to show you, also this was a really interesting example, H2AK119 ubiquinylation repressive mark. This was also showed at the same time, like 1 hour post-infection, there is clear ubiquinylation activities on histones could be observed. So this showed us that this is histone specific, not all the marks showing clear distinction, but we could identify on the course of infection the hot point where the shifts are there, which is for us now 1 and 3 hours almost, and then the other hours are like control, which give me the impression that this is spatial genomic rearrangement. There is a rearrangement occurs and it goes back to original state afterwards once virus is finishing transcription, and then it goes to replication state. So this was the methodological part that I want to share you. And also, I tested DNA, 5-methylcytosine and then 5-methyl-hydroxycytosine. Do they have specific effect on this methylation on DNA, and which type? And first, Luma assay, just total genomic DNA level. We did some tests, but here this observation was quite tricky. So many biological replicate. I think after 12 biological replicate, we could find this small change. As you can see, the number is very limited, but still logical because methylation should be above this percentage. So, and the sequencing data later on showed the similar levels. And again, early infection, there's some important demethylation occurs. And the labeling of confocal microscopy with using this 5-methyl-C antibody and 5-HMC antibodies. This was difficult antibodies, but we tried to make a quantification, and we show the result that there's really a weak staining, even controls, but a clear decrease of DNA methylation we observe here. And also in when we check 5-HMC antibody, we could see not important change in this methylation type. So also a virus targets specific methylation type on DNA in genome. So I also try another example of viruses, which is avian virus and avian cells. Here I see, for example, different pattern than the H1N1 and human. This is another host model. So it means that also this 5-MC in avian cells and avian influenza virus, which means that avian versus human influenza virus has some difference in targeting genome and then the methylation levels. So there is an host specificity also. So my observation on epigenetic marker, transient histone modification and DNA methylation events in the course of infection, in the early infection events are observed in the early 1 to 3 hours, between 1 to 3 hours, and these changes could be related to change in promoter methylation acetylation in certain genes. And afterwards, we did a study on cut and run to detect this regulated region in the same samples. But this is, of course, a nuclear infecting virus, and we use RSV, cytoplasm infecting virus, to see how they do the same nuclear reorganization stuff. And more or less, the model was selecting time points using microscopy. Then this selected time points, ChIP-seq, cut and run, cut and tag, or other methods could be useful. And then this is definitely a transient event in our model, and this we call genomic rearrangement. But now we can observe this histone modifications and methylation status. So if we can generate a generational cell passages with the infection, and then we can test different viruses and different bacteria-virus combinations or different microbial combinations, how they affect the systems, or also in vivo. So I think my time is finished. So I want to thank you, the collaborators from INSERM, Nathalie Bourgeon, and then Indra Bernard Delmas, and Dr. Herbein G, which we are collaborating on chip analysis, and of course in Consortium. And also I have a small announcement that there will be CLEPIC meeting next week. This year it was in June. It was in Szczecin, Poland. Next year it's also decided there, but open for any suggestions and combination with the COST symposium. And you can visit the CLEPIC website for updates. Thank you very much.
So thank you, Agah. If I understood you correctly, the quantifications you have shown are quantifications from immunofluorescence
Yes.
experiments, right?
Yeah.
This is always a little bit tricky because you have overlapping effects of changing the localization and changing the amount.
Yeah.
So the normal way to quantify these things, in addition, would be a Western blot to see whether you have also an upregulation, for example, of the H3K9 trimethyl in the hour after infection. So not only do it with immunofluorescence, because from my experience, and we do the same thing, this is always very critical.
Yeah, thank you for the suggestion. I will consider that. It's a good idea, actually. We can quantify easily and then measure the protein levels and put in equal proteins. And
that's a simple thing, just to have it as a...
Yeah, this antibody screening worked really troublesome because epigenetic-related, histone-related antibodies and DNA methylation-related antibodies, I tested so many brands and they don't work well. So, yeah, why I didn't do Western, I don't know, but... In the immunofluorescence, I focused because I want to see the specific pattern and the time point. So I will consider that Western blot because it will be very easy addition.
So given the first talk of the day, I mean this morning, I was just wondering whether the lysine 119 that you see to be changed has anything to do with enhancers and the 3D topology. So the integration. Do you see by microscopy some specific clusters of those sites in your nucleus?
A very good question, actually. That's why I want to go for super resolution microscopy level of observation on this same model, because this is too bright, so I couldn't see any further difference, but super resolution will help on that, I think, to see. But it's a very good suggestion. I was thinking of that.
Because related to the previous question, actually, you might have a change in the methyltransferase levels overall, but that doesn't prevent another layer of change at the 3D whatever. I mean, enhancers sites.
Thank you for your suggestion. Very good.